who will be, who has told me the secret title of this talk is Windows in Your Mind. Thank you very much to the organizers and for Gunnar for having a birthday and for giving us an opportunity to be here. Let's see if that's enough. Right, so the title is likely going to go on a survey paper later. It's snarky. Those of you who don't know the reference, it's for me and I need it. That better? Okay, so all right, no more time for title shenanigans. So I tried to make this as broadly accessible as possible. So there's going to be a little bit of neuroscience at the beginning that people who have seen me speak will be familiar with, but I think it's compelling, especially to topology. So I want to talk about something called a stimulus space, which is a model in neuroscience. And it goes back to the roots of sort of how we understand neural function. So this is an experiment, one of the very earliest electrophysiological experiments. So what you're going to see here is a stimulus presented to a cat, and there's an electrical probe in V1 in the primary visual cortex. And they're showing it this light bar. And they're recording, so I don't know if you can hear the pips in the audio. Maybe I can turn that up a bit. Oh, apparently not. Anyway, there are these very, very quiet pips that you might be able to hear. There. That's the neuron spike, the one neuron they're recording. So this was a groundbreaking Nobel Prize winning experiment. And what they're doing is finding what they call a receptive field for this neuron. It's a stimulus being presented to the animal, which the particular neuron is responsive to. So, and the existence of such a thing is sort of miraculous, right? It's one neuron in this cat's brain, and it's extremely active when the light bar is in a particular location at a particular angle. All right. So I won't show you the entire video because it goes on and on, but it's really neat to watch. So this is an experiment by Hubel and Weisel in the 50s. A class of neurons called simple cells. They respond preferentially to stimulus in a location at an orientation. And there are many of these in the visual cortex. So you can think about neurons in the visual cortex as parameterizing a space with these two different coordinates. A little bit later, we get a guy named John O'Keefe who did the same thing in a so that's primary visual cortex, that's direct visual stimulus. John O'Keefe, this is the picture that got me to do neuroscience, found deep in the rat's brain, the hippocampus, which is there, that there are these neurons, and each one of these colors is a different neuron spiking. And they're location sensitive, they're preferential to locations in the environment. So you get these, well, receptive fields, these areas where neurons spike. And man, you look at that picture, you come from a topology background, and it's hard not to say, I think that might be a cover of a topological space. And it turns out it is. We have really strong experimental evidence now that it's the intersection patterns there that matter. It's not the distances or the geometry, it's this and this intersect, these three intersect. So there's something topological about the information being represented in these systems. I'm going to pop my mask off. Sorry, it's hard to breathe in. So, okay, one more, and this one's, so O'Keefe won the Nobel Prize in the 70s. He won the Nobel Prize quite recently for work in the 70s, where he found these spatially localized place cells. One more, which is much more recent. And this one is just for the topology, because we're talking to topologists, you have to show this one. So this is an experiment by the Mosers, who have a lab in Norway. Niels Boss works with them quite closely. And so here we have a rat walking around in his environment, and they're recording an area called entorhinal cortex, which is just outside of hippocampus. This takes a second to get going, but it'll speed up here. So you can see the rat is, and that's just one cell they're recording. And you look at it and you say, man, that's just not as good as the place cell. Whatever it's doing in there is not, it's not spatially localized like I'd like it to be. But there's structure there, right? And if you stare at it for a long time, you see that it's not just kind of structure, it's a grid, it's what they call a grid cell. And grid cells, right, form this very regular sort of hexagonal looking thing in the environment. So these are 3 different examples I claim of the same kind of concept. So what are stimulus spaces? What are these things that I want to think about? And by the way, please stop me with questions. Any questions? All I know, nobody in this room is likely a neuroscientist outside of CAP. So. Okay, so we have neurons with localized responses. And so simple cells have localized responses to position and angle. And so I might think of that as R2 cross, say, RP1, if I were speaking to a room full of topologists. Place cells position in an environment. So these are spatially localized little spots in whatever environment the animal's in. It might be, a nice rectangle like this. If you're a bat, it's flying around in the air. You put it in an environment with obstacles, then you're going to have some holes. Grid cells are fun, a whole bunch of lattice points, but of course, everybody in here took a course on topology at some point or another, and you know that if I have a lattice, I can look at its fundamental domain. So let me just abstract this a little bit, right? I take my fundamental domain, glue the edges together like I want to, and then I do my gluing, and what do I end up with? End up with localization on a torus. So grid cells are spatially localized on a torus, which is fantastic, right? That's now we all get a big paycheck because we're the only people who know how to deal with these things. But it's also a neat torus because you have to twist it before you glue it together in order to get it to glue right. So it's not just the dumb torus, it's the cool twisted torus. Okay, and so there's a very, very cool paper from not 6 months ago now, where the Mosers and Ben Dunn, Phil's boss, a bunch of people got together and showed that you can robustly detect this structure in entorhinal cortex using topological methods. So it's a big win for us because those are some Nobel Prize winners and they're very, very happy with our methods. So, okay, so position on a torus. Okay, so what is the model here? This is a working definition. I don't think this is published anywhere, but this is how I think about this thing. So population of neurons, a bunch of neurons in the brain, has what we call a stimulus space coding scheme, right? If there's some metric space, which we'll call the stimulus space, it can be fancy. And the neurons are going to correspond to some points. So I think of them as sort of, embedded in the jello mold of the environment. And then every neuron gets a firing rate, and the firing rate falls off with distance to the corresponding point, right? So if I'm close to the point in the environment that I care about, my neuron's going crazy, but as I move away, it drops off. I don't have any real control, except I want those functions to be monotone decreasing because I don't want my system to be coding maliciously that I'm here and there. And then I'm going to take some threshold, some minimum firing rate, and say, if you're above this minimum firing rate, then that's the receptive field. That's the place where the neuron is active. And that's a good way to construct an open cover of my environment if I have enough neurons, or my stimulus space. So this is the conceptual model, and neuroscientists have been using this ever since the 50s from these first visual cortex experiments, but they haven't really been thinking about it in this way because they don't have, they haven't really had the mathematics right laying around. And it really took, the development of the tools of modern applied topology and geometry to make it reasonable to try to think about this model in terms of their data. So, and I look around this room, and half the people in it, their ideas have come into the work that we do here. Like, there's no way without this community being as great as it is that we could have, tried to do the things that we're trying to do. And I really think it's kind of amazing how fast we've come, because the other thing I remember is sitting in this room 9 years ago or whatever. Karina Kurto was giving the first talk on the work that we were doing on these ideas. So it's also, good place to be doing these things. So, okay, so under very mild hypotheses, if I assume this is a manifold or something, and I assume that there are enough of these functions and they're transverse in the right way or something, then what happens, I take the stimulus space, which is some abstract model of something in the world, and I put it into neural population space, which is the firing rates of my various neurons in my population. And that's an interesting embedding, right? We're very used to, in geometry, trying to find these minimal embeddings. This is wildly not minimal, 40,000, 100,000 neurons for a torus. So, but it must have some very special properties. And these are, if anybody is interested, we would love to know what those special properties are. The brain does it for a reason, right? There's no reason you would have this incredibly complicated machine doing these incredibly complicated energy using things to get a wild overrepresentation of something like this unless there was a really good reason. So understanding just the basic geometric properties of this embedding, I think, would be really interesting. Question? Okay, so neuroscientists have been very good at using knowledge of stimulus spaces to understand neural systems. Once they've got this model, so the three systems in the brain I claim that we know the most about are, in the order, vision, hippocampus, entorhinal cortex. Then you get things like motor cortex and auditory cortex and places like that. Auditory cortex is better understood, but it's sort of a one-dimensional, almost one-dimensional stimulus space, so you can cheat there. But we can do more, right? We can bring all of the cool tools of topology into the study of these things and learn things about them just from the observations. That's the thesis. And I guess it's a sort of an invitation. All of you guys know so much, and there's so much to be done here. So how do I, and really we, because I've never done any of this alone, how do we apply this formalism? So here's the data, right? I'm going to record a bunch of neurons firing for a while. I know way back in the back of my head that somewhere in there is an embedding of some kind of manifold. And what I would like to do is think about that model in the context of this data. So what I'm going to do today is based on using a cross time series similarity measure. It's like correlation, if you like, usually something a little more sophisticated, so you can think about the neural system. But I'm going to take a summary of that kind of data, and I'm going to use a symmetric matrix. And one great thing about symmetric matrices is everybody in the room knows what I'm going to do next. I'm going to put a filtration on it, and I'm going to call it I'm going to take the clique complex of the resulting filtered graph. I'm going to do persistent homology. That's been the game for quite a while. I'm not going to say Viator-Schrips complex because there's no reason in the world to think this thing is a metric. Also, it's a similarity measure right now, and that's not exactly like distances. So let me talk a bit about that. So suppose I have a simple stimulus space and environment, and I have these three receptive fields sitting in the environment. So I think I made this picture for the talk that Karina gave here 9 years ago, so glad I still have it. So the nerve theorem tells me that if I know the intersection patterns of these neurons, I can recover the topological structure of the stimulus space. And everybody goes, it's all very, I'm worried there might not be good intersections there, and you're right, it's all a lie. But it's almost a lie. There are two reasons why it might not be true. One is because I might have a bunch of noise, and the other one is because I might think it's a two-dimensional stimulus space, but I've just kind of intentionally projected the activity down onto the environment, but maybe there's more stuff going on, and my stimulus space is more complicated. We don't know that, but that's the standing assumption when we think about things like hippocampus and a lot of models. One reason why it's good to come at this from a zero knowledge, what can I come at, ground up mathematical perspective instead of a, let me tell you what this system is doing, now analyze it kind of. So, but let's assume that we're close enough so we can get away with it. Okay, so those are the center points of my three cells, or three receptive fields, and what you're going to say is, okay, I see there are distances between those, but I don't have access to the distances between those because all I have is this correlation data. right? But the animal's not teleporting around its environment, it's moving in a continuous fashion. So the two that are close together are going to be more highly correlated than the two that are than they're far apart. So the correlation, the ordering on the distances reflects the ordering on the correlations. So I just have to reverse the ordering on the correlations. I get a dissimilarity measure, and now I'm in business. So now I can play my persistent homology game. Okay, and so I can, and I will. And this is the paper that I was talking about. So, you know, top left is a picture that goes all the way back to Matt Kale and people doing Erdős-Rényi random clique complexes. And then if I take a geometric random graph in various dimensions, I get, and these are Betti curves, so they're you know, edge density versus cycle number. So it's like the dumbest possible summary statistic for persistent homology. So just think about adding up, right? Take the barcode or whatever and add up the number of bars that are alive at any given point. So you get these very, very stereotyped curves. And then of course you can take your place cells, you're recording from inside the brain of an animal and plug it through this system I just described to you. And it looks just like the geometric and nothing like the shuffle. And so you call it a day and go home. And it turns out that this works very well. You can detect the presence of stimulus spaces in neural activity just using basic persistent homology. And you see we've got Betti 1, Betti 2, and Betti 3 there. Of course, you can use Betti 4 and Betti 5, but by the time you get to Betti 3, you have a very, very sensitive geometry detector, essentially. Is there geometry in this system? Okay, so that's state of the art circa 2015. And then people started asking questions that were better, and I started trying to think about how you solve those problems. So the new question I want to talk about today is, okay, so remember I had that angular coordinate, right? And I think in Norway, Gunnar gave a talk about training a deep neural network so that it would do image reception. And the first thing you see in the first layer is this angular coordinate pop up, which is the same thing that V1 sees, right? So these angular coordinates are fundamental building blocks of what neural systems do, and they're everywhere. There's head direction cells, right, which of course are a pretty good circular coordinate system. Turns out that bats' head direction cells are tori for some reason. Anytime you have a pacemaker circuit, that's going to be a circular coordinate. These things are fundamental building blocks of neural coding, kinds of things you want the brain to keep track of. So the question is, I can find them, right? I just run persistent homology. The Moser lab and folks, they used technically persistent homology to determine there's a torus in the rhinal cortex, even though we verify that geometry is there. And suppose I have two of them. Suppose I know there's something in the environment circular and I see something in your brain, right? Or I am recording two distinct brain areas and there's a circular coordinate going in both. Can I tell you that they're the same? Can I tell you this is that and not this? Okay. In other words, can I assign semantics to neural activity based on things I already understand? That's really the question that we can answer using these topological tools that, as far as I know, otherwise is extremely difficult to do in any rigorous way. So why is it hard? Well, so here's my rat. I'm going to imagine it's running around in that circular maze. And I'm going to take a movie of it from overhead. Okay, that's going to be my input. And I'm going to record some neural activity again. And I go ahead and I compute, persistent homology. So in the video case, I've got a bunch of frames and I'm going to compute distances in some kind of pixel space. And so the animal, I've got two circles. One of them is the animal running around. What's the other one? Well, it turns out that the animal also, as it runs around, is turning. So I have an animal, I have that motion, right? And if it stands still, there's a perfectly good circle that I'm picking up in the aggregate. So I'm really doing that twisted torus we talked about earlier. Down here, I've just got one. So which one is it, right? And okay, so I go to the literature, and of course I go back to my initial topology experience. I say, well, I should get some functions, and I should compute some induced maps, and that'll be my thing. And then there's a problem, and the problem is I don't know the topological space here, and I don't know the topological space there. So in what circumstance should I expect to know a function between them? Right? How am I going to get a functor to apply to this induced map, because there's no ground truth in sight. And even if there was, why would I expect a function? Right? There might be things going on in that video that have nothing to do with what's going on down here, and I have no way of differentiating the two. Might be things in neural activity that have nothing to do with what's going on out there. No way to differentiate. So the best I might be able to hope for is a relation. Okay, so I can't just grab a thing. And then the other thing that you might think to do, right? This goes back to very, very early PDA papers. I'll take, the union of these two things, 2 point clouds, and I'll try to go through a zigzag. But the problem is, of course, there's no ambient space. So kind of out of luck there. Okay.
I got a bit confused.
Yeah.
So you have these different neurons that you're keeping track as the run moves. And now you do a correlation analysis to determine the distance between those, and that's the point cloud that you're computing.
No. So I have a point cloud in image space that gave me this core code. I have a point cloud in neuron space, right, which is just correlation between neural activity.
Yeah.
So that's totally behavior agnostic, right? So, right, the only thing I can ever hope to do is try to correlate this behavior against that behavior, right? Because I don't have any hope of anything more than that. But that's the data I can hope to get. So that's the data I'm going to have to use. So thank you. So I'm going to use a cross correlation, cross distance matrix. Okay, because that's the data that that's the only thing I can hope to observe in the lab. So yeah, so in here, I'm going to use P for the video and Q for the neural activity. And the reason for that is mostly because I tried to write video and neural activity everywhere, and it got kind of out of hand. And I should say that this solution, hiding down here is a citation to, this is all work. This part of this work is with Iris Yoon, who's here, and Rob Greist, who is unfortunately not. But yeah, everything here is really, I should say, 99% Iris. I was a passenger. So okay, so I've got this rectangular matrix. I know how to make a simplicial complex out of it. Right, I can make a Dowker complex of this matrix. What I'm going to do is I'm going to take, let's see, I have vertices corresponding to my rows and faces corresponding to my columns. Okay, and I'm going to threshold the matrix at some significance level. And then if I have, right, so and every collection of vertices I see, I'm going to call that a facet, and then I'll do downward closure. There might be some inclusion and stuff, but we just throw away the extra junk. Okay. And I secretly want to call this it's W because I want to call it a witness complex, but witness complexes have been used. So it's an argument that I will have offline with anyone who wants to have it. And then of course it's scale. So I'll let that vary and I get a filtration. Okay. So I just really briefly want to tell you why this is such a cool construction in case you are not familiar with the most important theorem, the reason it's called the Dowker complex. So here's a really small version of this thing, right? It's just a simplicial complex that I'm going to build from that little matrix using the construction I just described to you, right? It's got 5 vertices and 4 faces, and they correspond to the elements over there. But there's no reason in the world to think that I should have computed similarity in the direction I computed it in, right? That would be silly. Why should similarity be directional? So I could, have done this. I could have said, well, let's let the elements in Q be my vertices and Q be my faces. I'll get that simplicial complex. And you look at those and you think, well, those kind of look the same, but they're not really. But the cool thing about about this complex is this so-called Dowker's theorem. If I take the barycentric subdivision of both complexes, then if I just squint, I can see that I can fit the barycentric subdivision of this complex inside that complex. And what's happened, I took the barycentric subdivision, I got a vertex for every simplex in this complex. What are the simplices? They're my rows, or sorry, my columns, which are the vertices downstairs. So it turns out that it doesn't take much to get from there to a proof that these two things are quasi-isomorphic or even homotopy equivalent, I believe. And so there's a persistent homology functorial version of that theorem. So it doesn't matter which way I decide to measure similarity, which system is the input and which one's the output, I'm gonna get the same answer. So this is nice because it tells me I'm not going to get the wrong answer by choosing something that I have no information about. Okay, so I'll compute the persistent homology of that thing. I see a long bar. Cool, I'm done. Those two things are the same. Except that's not the question. The question was I have two barcodes and I want to compare the bars in them. And this is neither of those complexes. It's just some nice circle. Okay, well, what do I have to do? The problem is, what have I done though? I've taken my data, the cross similarity, and I've given myself a new simplicial complex with vertices P. I've got an old simplicial complex with vertices P, and it's one that I care about. I could have done this with Q, then I'd have two complexes with vertices Q. So now what I have to do is figure out how to move from one of these between two arbitrary simplicial complexes on the same vertex set in a sensible way. So,
I'm sorry.
Yeah.
When you say move, you mean create a map between them?
something like that.
Something like that.
Something like that.
Create a relation.
Create a relation.
All right.
Yeah, I'm never going to get anywhere near a map, unfortunately, in this brave new world of data. There will be a lot of maps involved, but none of them, unfortunately, will give me the answer I want. Yeah, I want to relate this thing. I want to use this thing that we've built out of this data to tell me which bars in the first one and which bars in the second one might be the same for whatever version of that sentence you want to give me. And I do, let me tell you which one I like. I like being deeply permissive. I want any possible answer that you might think is right to work for me. So I'm going to try to be as generous as I can. So I'm going to take two filtered complexes on the same set of vertices. So here's one, and it's some nice simple little complex. And you see it's got a cycle, and the cycle evolves, and then it dies. And it's got a bar. Okay, that's going to be the bar I'm interested in the first complex. And so I'll call that tau. And that's the last place it's alive. And I'm interested in that because that gives me the largest number of possible representatives for that cycle that I can possibly hope to have. Right. So like I said, I want to be my watchword is permissiveness. So if I'm interested in that class tau, that persistent class tau, and I start way over at the birth, well then maybe somebody else is thinking about that smaller representative. And I really should at some point in the lifetime be willing to call the smaller class right there at omega, that tight little square I should call it tau for some people. So I want to look there. That's where I think it would be best to try to make the assessment. Is this cycle the same as that cycle? So I've got these three representatives, and those are going to be, the things that I am allowed to think are tau. Okay, so now here's my other complex, and I've carefully constructed it in a terrible way. And the goal here is to make you say, well, if I just look at the same filtration index as I did before, then I'm probably going to want, I'm going to maybe try to minimize, I might try to minimize cycle length. That's something we like to do nowadays, right? Try to find a good generator. But if I do that at that late stage, I'm not going to think that they're the same. So I want to think about all of the possible representatives, because if I'm too choosy, I'm not going to match something which, I think one could very reasonably argue those two are the same cycle in many situations. And I will say, every parameter I'm telling you right now is choosable in the process. So if you say, well, actually, I don't want to be as permissive as you, you're certainly welcome not to do that. I don't know anything about the underlying science. I can't tell you about the neural system. And since I can't do that, I have to be as permissive as I can. We'll leave it to neuroscientists to tell me that I shouldn't have been. Okay. So yeah, I don't want to look at omega because I won't, I could try to get a map between those complexes like that. So what I want to do is I want to say, look, there are representatives of tau that appear on that bar. And that's the information I want to see. That's the thing I want to think about. So I want to find classes down here in the first persistent homology. That should be a P that intersect, well, at any T that intersect my tau. That's going to be, right, I'm on the same vertex set. So if I have a cycle that goes through the same set of simplices as one of my representatives, then that might be the right class. Might not be. I'll have to, then use more sophisticated methods to find out that I was wrong, but I want to give you a complete list of the possible choices. Okay, so I'm going to introduce new filtration. Those are my two simplicial complexes, and what I want to do is look for things that intersect that class tau. So what I'll do is I'll just take the complex at that critical parameter for tau, right, omega, and I'm going to intersect it with the other complex. What happens there? Well, now if the second complex, if sigma 2 is contractible by the end of its filtration, what's going to be left, right? As I go out this way, all that's going to be left are cycles that were alive, right? This becomes contractible. What's left here is just the stuff that was alive when tau was alive. So it's a copy, right? The infinite bars are a copy of the homology at that parameter. So what I'm going to do is I'm going to enumerate elements in sigma 2 at any given parameter. They're an image of that long bar, right? The bar that I care about. That bar is going to be the tau bar. So now I have a map, an actual map on homology. I can actually compute it, and it's going to tell me these are the things that correspond to, right, to the bar that corresponds to, right? Because that long bar is, there's one of those long bars is the tau class. Okay. So that gives me a method, a linear, basically I can now, I've got a linear system. I can ask the question. So, okay, so now I can do this thing. I can take any class on this complex on vertex set P. I pick up this homology, persistent homology class. I choose the parameter right before it dies. And now I look for all the things that sort of map through this fun zigzag of this new filtration. And that's going to give me a list of cycles over here that might have been that one. In other words, they intersect it at least one representative. Okay. So here are my three things. I want to know about that bar. And I should, by the way, say, everywhere I'm drawing bars, I'm going to draw some persistence diagrams in a couple minutes. I'm really thinking about the persistent module, right? Because there's all these change of basis issues. So I'm really thinking about a class in the persistence module in these pictures, but you don't want to see that picture. Nobody wants to see a picture of the persistence module with all that stuff. So that tau is really a homology class, persistent homology class. Okay. So I'm going to extend it over here onto this complex with the same vertex set. Then we apply Dowker's theorem, and boom, I'm on a complex with the other vertex set. And I'm going to take each one of the extensions I got here. There might be a lot of them, right? There might be many, many, many things that intersected that class, and I'll shoot them up there the same way. So I'm going to do the same process a second time to get them back over. So now what I've got is, in fact, a relation. That bar there through the data of the witness, or the Dowker complex, is related to a whole lot of potentially different looking bars up top. But I claim that it is an extremely permissive, and this is not unique in any way, right? There are probably other answers to this question, but this one gives you, I think, my money combinatorially the most permissive possible list, which you can then use to generate hypotheses. And in particular, if something doesn't show up, it'd be really hard for me to think of an argument that, according to the data, these two things are the same, right? Because we really tried to list everything that could possibly have touched any of the ways that you represent this class. Okay, so this is called the method of analogous bars, because the method of homologous bars would have been really bad. Does it work? All of this nonsense that you just told us how to do, is it useful for anything? So okay. So we had, I was going to show you a little video here, but it keeps choking the computer. So and also we're missing it. Okay, so these slides for some reason are not showing it perfectly. So I'm gonna think about a particular stimulus, and we're gonna build a neural network that's gonna react to that stimulus. Okay, in the simulation. And the stimulus is going to look like this. It's going to be like the visual cortex stuff I showed you. So we're going to show little localized gratings, but I'm going to let you run off the edge. So I'm going to do it on a torus. So you've got these little circle disks that have this sort of grating, and and you see there's the angle on the grating. That's one direction that we know the visual cortex is sensitive to. But then if I let the stimulus roll off this edge of your vision and come back for a little while, I'm going to get, you know, a connectivity between these two. Same with this and this. So we're going to teach your neural cortex for just a moment to be a 3 torus. So you've got 3 circular directions in the stimulus space. Oh, there we go. And so then we get, so we compute persistence homology of those videos, right? Like I told you, the pixel space. And I get 3 long bars. Great, perfect, exactly what we want to see. If we'd set it up like that and it hadn't worked out, and it didn't at first because there's some geometry issues when you have those gratings, then you have to fix it. Okay. And then I'm going to train some neurons that are putatively V1 neurons, and they're supposed to respond like the neurons in your visual cortex, right? So it's supposed to be direction and orientation sensitive, or location orientation sensitive. So the neurons have receptive fields that look kind of like this. And if we just brute force train those neurons to respond to individual location and direction stimuli, then we should also get a 3 torus inside the neural network, right? That should be the space it's representing. Each neuron represents a location on the 3 torus, and we forced it all to be this way. So we go ahead and compute persistent homology there. After we've trained this neural network, and lo and behold, 3 long bars. Great. And then, you know, proof is in the pudding. So we, okay, something strange is happening with slides. I apologize. I'm getting all sorts of mixed slide messages here. There we go. I don't know why that stuff keeps disappearing. Okay, the colors are the matches. So we apply this analogous bars method I told you about, and those long bars, along with, and I should say, these are what we call the primary, or we have some fun word for them. There are a lot of small bars that are potentially involved in these matches, but it's almost impossible to visualize those in any sensible way. So you should imagine a smattering of things down here for each one of these matches. But of course, that has to be the case, right? In any case, we get very, very clear, very, very strong witness complex. You know, this bar to this matches to this thing, and the witness complex matches to this thing. And of course, that's because we cheated and set everything up to work that way. But it does do what it's supposed to do in the complex of these messy simulations, which are like the neuroscientists do, right? These are spiking neural networks where we trained the neurons without any, you know, we trained them, of course, to do what we want them to do, but without like getting in and tweaking parameters. Okay, so I don't know why we're going to show this again. Whatever. Okay, so now we're going to test. That was the does it do exactly what it should do in sort of the most naive situation. So let me now take these spiking neurons here, the V1 neurons we just told you about, and train a subpopulation, a downstream visual system that's just supposed to pick out orientation. Okay. And let me pick out another one that's supposed to pick out direction. So, and this is not direction. This is information about a path on the torus, right? I'm moving that stimulus in a direction now. And there would be videos here, but I don't want to press my luck with whatever's going on with these slides. If I click on that, we may crash the machine. So these neurons, individual neurons, are recording the grading is moving in some direction, and there's nowhere in V1 that is explicitly represented by a neuron. It's represented by a time series of neural responses, right? This location, this location, this location, this location. In particular, none of those 3 S1 components, right, that I saw up there should come down here. Whereas up there, of course, we should pick out a particular one. Okay, so there are persistence diagrams, and the orientation neurons and the direction neurons do in fact have long bars. Great. That's right. I trained the neural networks. Iris trained the neural networks. I did not do that. And we get these nice long bars. And I don't know what's going on with these slides. Just really does not want to. Okay, there. Anyway, point is, the pink goes to the pink goes to the pink, as you might like in life, because that is, you can go all the way back to the stimulus and check those are the orientations, but we're only taking the input from the V1 neurons, so we aren't even referencing the behavior. And the direction neurons have an S1, but it's none of the S1s we've seen. So it is, you know, a computation and not just moving information. And we can tell it without, and the fact that it's not matched, like I said, is extremely strong. It tells you there's no interpretation of the correlations that would give you that match. Okay, so I'm almost done. I just have one quick thing, and this is highly, highly in progress. So I should say we tried it on data from animal recordings in multiple brain regions. Looks like a charm. We're very pleased with how it came out. That paper is forthcoming as soon as I manage to write it. So then work in progress here. So now that we know we can do this, Nico Schoencheck, who is also in the audience, is working with me on asking questions about, okay, so now I know I can track these things, which now gives me the power to ask the following question. So suppose I have a little feed-forward neural network, and over here I've encoded circular, maybe all sorts of different topological stimuli, and I've got a downstream system. What are the architectures for that network? What are the wiring diagrams that transmit this circle faithfully, but not that one? Right? When can I say, and how do I learn that? What are the learning rules that are biologically feasible and low energy that let me say, I want to let this circular coordinate through into this brain region so that I can have its information. Or maybe I want to do a process. I want to pick up those time series on the torus, right? That's what I actually want to pick up for you. So, and we're just getting off the ground here. We've just got our first simulations going. So this is, this next bit of the slide comes with a giant. I might tell you the opposite thing is true in 6 months asterisk, but we really believe that this seems right. So let's say I randomly wire it. I just choose completely uniformly random wiring between the neurons in the upstream population and downstream population. I train the upstream population, the input, to have a circle, because that's the easiest thing for me to track. And then I do what I told you I was going to do, right? I just push the neural activity through. I compute the correlations, cross correlations, blah, blah, blah. Apply my method. And what do I see? I see a long bar in the barcode and a long bar downstream. So my random network might have transmitted the circle. However, at this time, we are not getting a match between the top circle and the bottom circle. There is a circle being represented downstream, but it's not that one. No idea what that means. If you have a hypothesis what that means, I would love to have a conversation. But somehow, a different circular coordinate arises from the goo of a randomly wired feed-forward neural network that isn't the one that went in. So I'm deeply pleased with this picture. I think this is one of the coolest pictures I've seen in a long time, because it tells you the brain, it might tell you that the brain spontaneously generates circular coordinates that you can then wire things into. Right? That would be like that would make sense because we need those, right? We use them all the time for representing the real world. Or this could be a bug in our code. So, coin toss, either this is really cool or it's really cool for a different reason because we found the bug in our code. So I want to thank a lot of people. None of this is possible without every single person on this slide, about half of whom are in the room. This is my dog Scout, who has a torus that she likes to bring to me when I'm frustrated with topology. Of course, I have to thank my sponsors, and I'd be happy to take any questions.
What happens if you feed a signal in that doesn't have a circle? We still get a circle out?
That's a great question. I have no idea. So the question is, if we take that same randomly wired network and we shove noise in, we get a circle out. And we just got this is from last Wednesday, I believe, is when Nico sent me this picture. So we finally have all of the simulation code and everything, and the extension method up and running again. So yeah, we're fingers crossed. I'll be able to answer that for you in a few weeks.
Can you say a little more about what you mean that they're not the same circle? In other words, quite like, do they sit in some common domain? But
yeah, that's a great question. So I have neural activity space in the domain and neural activity space in the codomain.
Yeah.
And then I have I've got this cross similarity matrix. And what I mean is, right, when I push this thing down to the witness complex via that extension method,
yeah,
it shares so it doesn't hit this long bar. So the image is maybe that short bar, maybe it's trivial, but it's not hitting the right homology class. And as a result, even though this long bar I believe, and I don't think we've checked this, but this is the most sensible possible interpretation, even though this long bar and this long bar are the same. In the sense that, I go from the Dowker dual of the original complex down to here, right? So these two do share a representative, but these two don't share a
So
representative.
can I think of it as, like there's some ambient space on the target side,
Mhm.
which is bigger than any one circle or something, and that maybe there's some basins in there, and this is okay. Is that a fair?
Yeah, I would guess, just based on knowing, right? So the geometry of these things are these sorts of, so the geometry of these perceptron feed-forward neural networks, right, I have, you think about neural activity space as being the input neuron space, and then there's a hyper or a half space in them for every output neuron. And the weighting vector there is the normal vector. And so the idea is that if I want to capture the topology of that circle, I somehow have to not cone off the region. I have to have a collection of regions that are sort of witnessing the circle via the nerve theorem, but not cone it off. And my guess is what happens when you're running a randomly wire with probability when you cone. But nonetheless, you end up with chunks of this very large dimensional space, which, because there aren't enough neurons to give you a full cover, there is somewhere where you could get a circle.
So are you going to try wirings that are not random?
Yes. Yeah. This was just like out the box. Let's see what happens with because, the base hypothesis in all of neuroscience is everything in the brain is completely random and there's no energy that went into it. And right, if you ask neuroscientists what's happening, and you say, I don't think that's right. And they say, we don't either, but until you can prove to me it's not true, then that's what I'm going to say to you, about everything that you do. Of course they don't believe it, but they need you to prove this thing is not random before they're going to believe it's not random. We find these posters of like, I scrambled the neural activity and it gave me the same PCA, and so these things are all random. No one believes it, but it's like their base mindset. So you always have to. This is kind of a dumb thing.
So the method that you use to compare and make analogy between different bars, what would be the more abstract setting in which it could be used?
I don't know. That's a great question. So what are we doing really? We're saying, I believe that so in my head, there is geometry, right? Let me say the following thing. The stimulus space model has the following implication. Suppose I have a stimulus space in this region and a stimulus space in this region, they're wired together, right? Instead of thinking them as separate stimulus spaces, I could just mush the neural populations together and get one big stimulus space, which is, naively maybe the product of the stimulus spaces, but probably not. Right? It's probably some kind of twisted, who knows what, just a bigger space that contains, these circles and those circles. And so we're trying to think about something about the geometry. There's something in the geometry of these two spaces, but I don't know what it is that relates the two. And I'm probably wandering too specifically into neuroscience again, but yeah, I guess I feel like I don't know how to answer the question, how do you generalize this? Because I don't know what I would want to generalize it. It's essentially just a matter of trying to answer the question in the absence of a function, which I cannot possibly have observed. How do I ever possibly come up with a map on homology? I think we haven't proved a lot about these things. And the reason we haven't proved a lot about these things is because I did not a deep search, but not a cursory search looking for, the category of topological spaces and relations, and didn't find a lot. But I think that is the right setting to ask these questions, right? You say, okay, right? So Dowker's theorem is about this very discrete setting, but I could, so if you think about the proof of Dowker's theorem, one way to think about it is you've got a join, right, of these two spaces, and you're sort of yanking homology classes through, and you prove it, you go through sort of an intermediate space that lets you prove that they're all represented. So if I just forget that intermediate space and I think about joins as being, I join open sets over here with open sets over here, and I take Right? That's sort of a continuous Docker construction in some sense. And that's very, very vague. I don't mean to tell you that I know what I'm talking about here. I could imagine that you might, right? But if we want to prove things about this kind of construction, I think that's where you do it, right? You look at this category where your maps are relations. Okay, what can I say about maps on homology? And you try to prove things about bottleneck distance, right, and Wasserstein distance, things like that. Very often, one of the tools you'll look at is, I have a point set here and a point set here, and I'm going to take this relation and I'm going to sort of imagine coning off all the points that are close enough here, and that's why the homology is close. And that's the same kind of in the same spirit. Right? I have these topological spaces, and I'm imagining sort of trivializing local parts of them in a way which is consistent with the homology I observed. So I guess that would be where I would, and that's kind of, I have some scribbled notes along those lines, but that's where I imagine generalization starting, and where you would actually prove any really useful stability properties or anything else that should be here by generalizing.
Just say again, how do you get do the comparison of P and Q if they're on like very different scales?
Right. So okay. So the devil is always in the details. Right. So I've got these two systems P and Q. Whatever. I'll just stop here. Okay. So I've got my systems P and Q. I can measure internal similarity here and internal similarity there, and I'm leaving it up to the person doing the experiment to tell me what that means. I'm also leaving it up to the person doing the experiment to tell me how to measure cross similarity. If they're just time series, right, then that's maybe, if they're neural time series, I might want to put a delay in because there's transit time between neurons. If it's a video and a time series, I might just use correlation again with a temporal delay or some kind of smear. But, I think of this as a general tool for telling you these things might be the same. And so whatever the things are, there's got to be a domain expert in the room to tell me how to actually interpret both what I'm putting in and what I'm taking out. If I don't have that, all is lost. And you should never try to use these tools unsupervised.
All right. Let's thank the speaker again. And our next talk will be in 9 minutes.